Hello, and welcome to What is a Journal Club? I'm your host, Mr. Johnny Hopkins, and today we'll be covering something called side chain interactions governing the pairing of half-cystine residues in ribonuclease, as done by Edgar Haber and Christian B. Anfinsen. This is a very, very famous paper. It's from the Journal of Biological Chemistry, volume 237, number 6, June 1962. It was printed in the USA, believe it or not. So, essentially, this is a very important paper in the history of biology and chemistry, and biochemistry in general, that focused on, in fact, the refolding of this protein called ribonuclease. And what does ribonuclease do? It's also known as RNase. It chops up RNA, essentially. And so, effectively, we're going to just go through like an introduction abstract, go through the methods, then talk through the results, and then, what does it mean? Go through a little bit of a discussion. And so, our ribonuclease is also known as RNase. And what they had is what ribonuclease has is 8 sulfhydryl groups, 8 SH groups, which means it has 8 cysteine residues. So what happens in the cell is typically a protein will fold and then cysteines will latch onto each other and form a disulfide bond. So effectively, those SHs will go and collapse into just an S and an S, like here. And so that helps form the structure together and keep its enzymatic function. And so effectively, we have 105 possible arrangements of all these guys interlocking because of handshake theorem and all that fun stuff. So then, if we have 105 possible arrangements, how is it able to actually achieve its function? Is it if we presume that if we have an incorrect pairing of these sulfhydryl groups, that means we probably have not good function of this protein, if even any function at all. But we have to have the 1 in 105, we have to have that 1% possibility. And what happens? What drives this process? And so that's what Haber and Anfinsen decided to go and set out to do. So in this method, they essentially have a protein, ribonuclease, RNase, and it's got these 8 sulfhydryl groups, and it's in a native form. They believe purchased it from Sigma Chemical Company with the lot number R60B204. It's coming on the test. But so they purchased this guy because it was very easy to purchase and study, and it was also very well studied and well known, especially like they know the entire sequence. They know that there are 8 cysteine residues, in fact. So it's a well-studied protein, so they take this protein, they reduce it, and so then it becomes essentially just a long peptide chain. It's got it's got all this removed sulfhydryl groups. It's just all, they're all free cysteines bound to each other. There's no disulfide bonds, as well as it's just kind of floating around, just being a free wiggly boy. So what they did to reduce it was put in some ethanol solution in the presence of 8 molar urea. And so then we got a wiggly boy, peptide boy, and then you oxidize it back and they change the pH and the reagents. And they tried to see whether or not this structure was the same as this structure by just measuring the activity. And the way that they just did that is by they pretty much purified this protein. We'll not get into any of the discussion of that. We'll just believe them. They purified it out, put it into a solution with RNA, and then measured the activity by measuring the absorbance of the RNA itself. So, of the RNase and the RNA. So, that's essentially what they did. And so, effectively, the main crux of all these results I'm going to talk about are just the change in the pH and the reagents. So, first of all, so essentially, the majority is just this step right here. So, first of all, they reduce it and then go into table 1 real quick. And table 1 describes the effects of what happens when we add in, when we make a reagent, urea, to oxidize the solution back from this guy to that guy. When we oxidize and we add urea, when we have a small amount of urea, 0.25, we can get a we can regain the activity of 100%. And then when we add more and more urea, we reduce the activity that is regained by this oxidation step. Same thing happens with guanidinium chloride, except guanidinium chloride is a stronger material than urea in this instance, and so we have 1 molar solution, we only get 88% activity recovery, and then we go on, we get only 4. We get less than 1% at 4 molar concentration during the oxidation process. So one thing to quickly notice that during this process, when we go to the oxidized form, they make sure that there are no free sulfhydryl groups in the solution, that it was all entirely just meshing. That everything had a disulfide bond, there was nothing. The process had finished. We went completely oxidized the solution. Now we're going to go into figure 2, where figure 1 effectively just goes over the rates of the oxidation process of these sulfhydryl groups, so it's not as critical as figure 2, where they looked at how does this oxidation process occur with the change in pH. So when you oxidize it, you can see how the recovery goes with the oxidation. So what they just did essentially is change the pH. I should have changed, but anyway, label here. This is 8. About here is 2. So you go on, you go on, you go on. This is about 6. 4. So essentially it's just use sharp change from here to here. And so quickly noting, this sigmoid curve is the native, and the dots are the oxidized. So essentially, when we just instead have this native RNase right over here, and we just measure its activity, so that's just all that this is. When we change the pH, we see very low activity at low pH, and very high activity at, let's say 100% activity at pH 8. And we see the sharp shift from 4 to 6 in general. And if you typically in the cell, you don't even go below 4.5% pH in the cell, and that's usually like 4.5 is more around like the endosome and lysosome compartments of the cell. So RNase is still able to actively perform in those sort of compartments and organelles, but typically at like this pH of 6.5, we see a significant high amount of activity, such that it's pretty indistinguishable from just the native at a really, really high state pH. So then these dots essentially show that when we reduce it, now we oxidize it back and we measure the activity, at these points, we pretty much see the exact same enzymatic activity. In fact, we actually see a higher percent of regain typically than the native wild type, which could just be chemical error and analysis problems, or it could just mean that somehow it's just magically doing it. But essentially we'll just say that when we oxidize the solution in these pHs, we see a regain of activity when we measure it in these pH. So now continuing on, we have figure 3. This is essentially just what they looked at to see how the spectra of RNase is when you have in the reduced versus the oxidized form with 8 molar urea. And so when we have the reduced form, we see actually a little bit of a dip or an indiscriminacy around 2,800 angstroms in lambda at the wavelength of the absorbance. So this is an absorbance measurement. So if you remember Beer's law, absorbance equals epsilon I think. Yeah. So essentially it's just absorbance is equal to the absorbance coefficient, the times the concentration times the length of your media. So when you look at that and get the absorbance, and you can see how the absorbance changes. Essentially, when you have the oxidized form, it's got the exact same shape, just a lower absorbance, whereas you get this slight change in the reduced form. So going on from there, that's essentially some of the data that they collected, what most of the data that did looks like. I thought it would be important to show that. And then we go into table 4. And table 4 is actually a really interesting thing, I would say. So trypsin, they did some trypsin digestion. So what is trypsin? And trypsin is a protease. Trypsin cleaves proteins at different positions based on the sequence. So when it's able to sense a sequence or a motif in a protein, it will cleave that part, and then those peptides, those cleaved ones will go out and be fully degraded into their constituent amino acids, or go into MHC complexes. Don't have to worry about that, but so they get cleaved, and then they go and they can recycle the protein. So they looked at trypsin digesting this guy, and when you have the native, just the native regular purchased from Sigma Aldrich RNase, there are no bonds cleaved when you have it that way. But then when you reduce it and you make it into a full-length peptide, that means you have 11 bonds cleaved. So that means you have 12 different peptides for this entire RNase, whereas when you have native, you get just the same old, same old. So when they continue on, when they oxidize in a buffer, what they saw was just a buffer that gets that when you have pretty much a significant amount of activity, you see practically no bonds cleaved. You get less than 1 on average, which means they have full reactivity typically in general. And that means the oxidized one and the native one are practically one and the same, because they are both undegradable by trypsin. And they are, And so when you have urea, you get 9 bonds cleaved, which means you have 10 peptides, and guanidine chloride, 11, oxidized in 3.5 pH, which is right over here with very, very low activity. You get 11 bonds cleaved. So as stated before, the more stable the structure, the less likely trypsin is to be able to degrade it. So, and that means that these guys have have less ability to regain into their native form, which is the most compact and strongest form. So they're able to be cleaved better. Continuing on, we have table 5 finally, and so effectively what happened, what this is just a very similar experiment to previously, where they oxidized in different components. So they oxidized the RNase at a pH of 5, urea and guanidine chloride, and they saw that the activity, once they just did that, they took an aliquot, purified it, measured the activity in 7.11, practically nothing. Then they incubated it in 24 hours at pH of 8, and they saw pretty much the exact same activity. So there's like nothing that happened. But then when they put it in the excess of the ethanol derivative thingy, and they saw a significant increase in the activity after 24 hours of this incubation time. And that is the thing that was used to reduce the solution itself. And it was a 100-fold excess of beta-catenol. So doing that, they're able to see that you can do 24 hours of a incubation in a unnatural environment, but then still the protein will refold and go back to its native state afterwards and have a significant amount of activity and functionality, even though it was in this horrible environment that it should not have been in the first place. So that's effectively all the results that I'd like to cover for this paper. So what does this all mean essentially? You got the mixing in urea, guanidine chloride, and all this other stuff, and just trying to like, hey, what's going on? And it's kind of difficult when you just look at these small details, because these details kind of drive into these tall weeds that aren't quite as easy to tell in the story. But if we just think of it this way, we reduce it, and then we oxidize it, and then measure the activity. So we make it into a wiggly boy, so we tear apart, we just pull from end to end this protein from end to end, so no longer it's just this long peptide chain. It's got nothing interesting going on about it. And then we let it refold, and then we let it reform its cysteine disulfide bonds. So we pull it apart and then just let it refold. And then at the end, you get the rate gain of activity. You get the same structure back. You go from pulling it apart and then it folds back in. You pull it apart again, it folds back in. And the way it folds back in is the same every single time in physiologically relevant situations, such as low urea concentration, whereas if you have a high urea concentration, which is also something that will unfold proteins, it will unfold the protein. But when you have just a general physiological condition, you pull it apart, it folds back in the exact same way, and it goes back to its original functionality. So, I'd like to leave you with just the ending remarks of this paper. When the incorrectly oxidized derivatives were dissolved under conditions compatible with disulfide interchange, pH 8 in the presence of sulfhydryl compounds, they regained a large fraction of the theoretical activity. These results suggest that the native molecule is the most stable configuration, thermodynamically speaking, and is the major force in the correct pairing of sulfhydryl groups in disulfide linkage, is the concerted interaction of side chain functional groups distributed along the primary sequence. It's a little bit of a mouthful, but as I said, it just means you pull it apart, it comes back together the exact same way, and these disulfide bonds come back together, and it's a beautiful mixture, and that is part of the, that is essentially just all that led to this Anfinsen's dogma that the protein sequence defines the structure. That the sequence is able to determine what fold it goes into, which determines its functionality and activity inside the cell. So that's all I wanted to cover for this paper, side chain interactions governing the pairing of half-cystine residues in ribonuclease by Haber and Anfinsen. Thank you.